So hello everyone, in this video we'll study an important aspect of pharmacodynamics, that is receptors. So we know receptors are very important to exert the pharmacological or therapeutic action of the natural neurotransmitters present in the body, as well as to exert a therapeutic action of the drugs that we take to treat or manage many disorders and diseases. So first of all, what is pharmacodynamics? Pharmacodynamics is defined as a branch of pharmacology that deals with the study of effects of drugs and the mechanism of action of the drugs. That is, what the drugs do to the body and how the drugs produce that action. So what the drug do to the body is nothing but the effect of the drug or the pharmacological action of the drug, or you can also call it as therapeutic action or therapeutic effect of the drug. And how the drugs show that action is nothing but the mechanism of action of drugs. So in order to understand the pharmacodynamics of a drug, we need to understand the relationship between the action and effect of the drug. We need to understand the relationship between the dose and effect of the drug, and also the interactions of the drug with other drugs and also with the food. So basically, and coming to principles of drug action, what many drugs that we take, all the actions produced by different kinds of drugs that we have will come under either stimulation, depression, irritation, replacement, or cytotoxicity. For example, adrenaline stimulates heart. Acetylcholine depresses the SA node of the heart. Certain drugs are producing inflammation and redness and pain to the cells, a kind of hypersensitivity reaction or inflammation kind of reaction. So that will come under irritation reaction. Certain drugs we use to replace, we use it as a replacement to compensate the diminished levels in the body. For example, we use vitamins. If the natural vitamin levels in the body are lower than the normal range, if they are not sufficient to maintain normal physiological processes, then we need to supplementally take those constituents. For example, we take calcium, we take vitamins, vitamin E and vitamin C. In Parkinson, there are very decreased levels of dopamine, so we supplementally take levodopa. So all those actions of such kind of drugs will come under replacement kind of reaction. Now the last kind of action of a drug is cytotoxicity. Now we require we require cytotoxic action against two kinds of cells. One is microbial cells and the other is cancerous cells. In case of infections, we need to kill the microbes in order to remove them from the body. So there we need cytotoxic action against the microbes. And we also need cytotoxic action against cancerous cells in order to kill them and reduce their number. So whatever drugs that we use to treat various disorders and diseases and infections, their actions will come under one or the other of these five actions, that is either stimulation, either depression or irritation, or we use it as a replacement therapy, or we use it to exert a toxic action against either microbes or cancerous cells. So then the question is how these drugs produce these pharmacological or therapeutic actions. Now, many of the drugs produce their pharmacological or therapeutic action by acting on a receptor. Now, this receptor is not only important to exert the therapeutic action of a drug, but it is also important for exerting a physiological reaction of the natural neurotransmitter or hormone present in the body. For example, the natural adrenaline in our body shows its action by acting on its alpha-beta receptors. Similarly, natural acetylcholine present in our body shows its action by acting on cholinergic receptors. So receptors are important to show both the therapeutic action of the drug as well as the physiological reactions of the natural neurotransmitters and hormones present in our body. Now, when a drug activates the receptor, there is either stimulation or inhibition of an enzyme. There is either opening or closing of some ion channel, or there is either facilitation of the transport of certain constituent across a transporter, or inhibition of the transport of the substance. So ultimately, that is why these enzymes, ion channels and transporters are called as effectors, because when a drug activates the receptor, ultimately there is some change happening in the enzymes, ion channels and transporters, and a pharmacological action or therapeutic action is being produced. Most of the drugs act by activating receptor. There are certain drugs which directly show their action by either activating or inhibiting the enzyme, either by directly opening or closing the ion channel, or directly by activating or opening the transporter, or by blocking the transporter. Now, apart from this, there are certain drugs which show their action by their physical or chemical property. For example, we have bulk laxatives. They act by increasing the physical mass in the large intestine, which helps in defecation. So they are used in constipation. We have charcoal, which acts by its adsorption property. It absorbs all the chemical constituents present around itself. That is why it is used in poisoning, to absorb poisons. Then we have radioactive iodine. Through its radioactive property, it helps in killing the thyroid follicular cells to treat certain thyroid disorders. But we have very few drugs which act by their physical or chemical property, but most of the drugs that are present act by activating the receptor or either inhibiting the receptor. And ultimately, when a receptor is activated, there is some alteration in the enzyme, ion channel or transporter or any other substrate, and the therapeutic action is produced. So the question is, what happens when a drug activates the receptor? So there are different kinds of receptors like G protein coupled receptors, receptors linked with ion channels, enzyme linked receptors, and receptors regulating gene expression. So in this video, we will see what are G protein coupled receptors. What happens when a drug activates G protein coupled receptors, what mechanisms or which pathways get activated, and how a pharmacological action is produced. Because many of the receptors in our body are G protein coupled receptors. So what is a G protein coupled receptor? First we'll study about its structure. It is a transmembrane receptor which is present on the cell surface or cell membrane, and it has two sites. One is ligand binding site, which is present on the extracellular side, and the other is G protein binding site, which is present intracellularly, that is inside the cell. So inside the cell, in the intracellularly, this receptor is bound to a G protein. That's why the name G protein coupled receptor. And also along with that G protein, there are three subunits, that is alpha, beta, and gamma. When the receptor is in inactivated state, that is when there is no drug bound to the receptor on its ligand binding site on the extracellular surface, it is an inactivated state, and at that time the receptor is GDP bound, GDP, guanosine diphosphate bound, and the alpha, beta and gamma subunits are together. But when a drug activates the receptor becomes GTP bound, and the alpha subunit dissociates from the beta and gamma subunits. So this is about the structure of the G protein coupled receptor. There are how many types of G proteins? There are four. One is GS, GI, G0 and GQ. Certain receptors are bound to GS protein, certain receptors GI, certain to G0 and certain to GQ. Now what happens when a drug binds to the receptor? Now, depending upon which protein is coupled to that particular activated receptor, one particular pathway gets activated and the pharmacological action is produced. So one by one we'll see what happens when a particular G protein gets activated. So first we'll study about what happens when GS protein gets activated. So if a drug activates a receptor which is bound to GS protein, a pathway called as adenyl cyclase CAMP pathway gets activated. For example, you can see in the diagram here, when a drug binds to GS protein coupled receptor, the GS protein gets activated. Activation of GS protein leads to activation of adenyl cyclase. It is an enzyme. This activation of adenyl cyclase, as the name itself indicates, it converts the ATP into cyclic AMP. Now this leads to increased intracellular concentration of CAMP, and which in turn leads to activation of protein kinase A. Protein kinase A causes the phosphorylation of different kinds of transporters, enzymes, and proteins and substrates, and that produces various kinds of reactions like contraction in the heart, relaxation in the smooth muscle, inhibition of secretion, lipolysis, glycogenolysis, etc. Apart from the activation of protein kinase A, when GS protein gets activated, there is also opening of calcium channels leading to increased calcium ion concentration inside the cell, which helps in contractility. So this is about GS protein. Whenever a drug activates a receptor which is coupled to G protein, adenyl cyclase CAMP pathway gets activated and a pharmacological action is produced. So this is the mechanism of action. Now what happens when GQ protein gets activated? Consider a receptor which is coupled to GQ protein. Now for example, you can see in the picture here, consider an example of a drug we have that is histamine. So if histamine is binding to this receptor and this receptor is coupled to GQ, so this receptor gets activated and the GQ protein gets activated. Activation of GQ protein will activate enzyme phospholipase C. This phospholipase C causes the hydrolysis of PIP2 into IP3 and DAG. PIP2 is phosphatidylinositol biphosphate, which leads to the production of IP3 and DAG, that is inositol triphosphate and diacylglycerol. Now IP3 is responsible for release of calcium from the calcium stores, and DAG, that is diacylglycerol, is responsible for the activation of protein kinase C. When protein kinase C gets activated, again it causes the phosphorylation of various kinds of substrates, that is enzymes, transporters and proteins, and it produces different kind of actions like contraction, stimulation of secretion, stimulation of neurotransmission, metabolism, cell proliferation, etc. So this is about GS and GQ protein. Now what happens when GI protein gets activated? Or more specifically, if we start from the first step, what happens when a drug activates a receptor which is bound to GI protein? What happens is the adenyl cyclase pathway and the phospholipase C pathway are inhibited, that is CAMP concentration decreases and IP3-DAG concentration decreases, and all the opposite actions are produced. And what happens when G0 protein gets activated? There is inhibition of calcium channels. So basically, to put up in a summary, when GS and GQ coupled receptors get activated, stimulation kind of reactions are produced. Not always, sometimes inhibition kind of reactions can also be produced, but usually these are called as stimulatory. And when GI and G0 protein get activated, there are inhibitory or relaxation kind of actions are produced. So this is about the mechanism or the pathways or the secondary messengers that get activated when GS, GQ, GI, G0 proteins get activated. So this is about secondary messengers. In case of adenyl cyclase pathway, who is our secondary messenger? It is CAMP. It is a secondary messenger. It is acting as a messenger and then the pharmacological actions are producing. Similarly, in phospholipase C pathway, our secondary messengers are IP3 and DAG. But sometimes, without the involvement of these secondary messengers, there are pharmacological actions produced. For example, when that is called as direct channel regulation. For example, when GS protein gets activated, there is opening of calcium channels in the cardiac muscles and in the skeletal muscles. Similarly, when the GI and G0 protein get activated, there is opening of potassium channels in the cardiac muscles and smooth muscles. So this is how G protein coupled receptors act, or this is what happens when a drug activates G protein coupled receptors. Depending upon which protein is bound to that activated receptor, a particular pathway gets activated and the pharmacological action is produced. So this is about G protein coupled receptors, which is a large family of receptors, and many of the drugs or produce their action by activating G protein coupled receptors. So thank you.